All right, in this video, we are going to take a look at the cell cycle, just a general overview. And you should know at this point that there are three major phases. Interphase, and interphase is made up of G0, which is where the cell is resting. In G1, the cell is preparing to grow, so getting larger in size. In the S phase of interphase, DNA replicates. And then last but not least, in G2, the cell grows a little bit more and gets ready to enter into mitosis, gets ready to start dividing up the chromosomes that it replicated in the S phase. During mitosis, down here, you can see that, we will see that what happens is the chromosomes that were replicated in the S phase of interphase are now divided. So the cell will go through the process of taking the duplicates and splitting them in half so that we can get to the last phase of the cell cycle, which is cytokinesis. In cytokinesis, you see cyto again. This is dividing the cell and the cytoplasm. So that's the only thing that really happens in cytokinesis is that final split. The cell has replicated the DNA in the S phase of interphase. We've split all of that DNA up in mitosis, and now we have to take that single cell and divide it in half so that we end up with two identical cells. That's the whole purpose of the cell cycle, is to end up with two genetically identical cells. The cell cycle is just, as I kind of just went through, an orderly sequence of events that starts with the cell replicating its DNA, dividing that DNA, and then dividing the cell. So it's a very orderly process. If the cell's not actively in the cell cycle, then it's in G0, where it's just resting until it receives some type of signal that it's needed to replicate, and then it will enter back into the cell cycle. The cell cycle, as I said before, results in two new identical daughter cells. They are genetically the same. The cell cycle allows us to replace worn out or dead cells. For example, your skin cells are always dying. And as they die and you come in contact with like your clothes or your sheets, or you brush up against something or someone, those cells are rubbed off. And if you didn't replace those cells constantly, then you would end up wearing away all of your skin. So the cell cycle helps in the process of replicating skin cells, making sure that they're replaced as they die. In addition, if you've ever had a sunburn, you've damaged all of those skin cells at the top, and when they peel off, they're dead, and your body's actively replacing those dead cells from the bottom up. Also, for the cell cycle, one of the roles is growth. Some stuff never stops growing, like your hair and your nails. So those have to be replaced over time. Also, to maintain proper surface area to volume ratio. The whole purpose of this is so that the cell doesn't get too big, so that it becomes inefficient in terms of transporting materials in, out, or around the cell. So you want the cell to be not too big and not too small so that it can move whatever it needs into to where it needs to go. And then more importantly too, you can move those wastes that are produced out of the cell. Cell reproduction is also known as cell division. So if you have a cell that you are just replicating that cell, it is also called cell division. So as we've talked about before, there are two types of reproduction: asexual reproduction that only has one parent involved, so no egg or sperm. And when this organism divides, it makes a copy of itself and its DNA. So there is very little genetic diversity from parent to offspring in asexual reproduction. In sexual reproduction, though, there are two parents. An egg is fertilized by a sperm, and in this instance, since you have two parents, you have two sets of genes that are being united, and as a result, you end up with quite a bit of genetic diversity. I mean, just look around the room, look as you walk through the hallways, you see lots of genetic diversities among humans, and humans reproduce sexually. All right, so let's talk a little bit about the types of asexual reproduction. Binary fission, bi means two, and fission means split. You should have learned that in physical science. And you can see this is an amoeba and just a regular cell. You can see that you start by replicating the nucleus or dividing the nucleus, I should say. And you can see that he's just starting to pinch in half. In picture 3, you've got a little cell furrow that's starting to occur to divide the cell, but your nuclei have already divided. 4 and 5 continue that split of the cell and the cytoplasm. You can see that starting to and continuing here. And then in picture 6, you have two cells. So binary, bi means two, and fission means split. So we're going to take one cell and we're going to split it into two cells in this in binary fission. Binary fission also occurs in prokaryotes. You see the exact same picture with a bacteria cell here. The green strand that you see right here is the original DNA. And then in the second picture, you can see that you have a strand of red and purple DNA. All the artist has done is shown like the pink may be the original DNA, and then the purple is the replicated DNA. You can also see then in the next image, you're starting to create that cell furrow. And then at the bottom, again, remember, this is binary fission. So we end up with two identical cells at the bottom, all right? So prokaryotes do this, even though they don't have a nucleus. They undergo binary fission, but there are some nucleated organisms, like you just saw in the previous slide. A lot of single-cell organisms do binary fission as well. The mitochondria in your cells divide like this. And as we've talked about before, this is a piece of evidence to support the endosymbiotic theory. In regeneration, you can see this larger arm on this sea star, and that's the original arm. And you can see that he has been able to regenerate the rest of his body from that single piece. So anytime a sea star is cut up, if you would have cut this into 5 pieces, you'd have ended up with 5 new organisms. So regeneration in the context of asexual reproduction allows an organism to recreate itself from just a piece. Now, I'm sure that quite a few of you are like, oh, reptiles do this. I've seen that when they lose their tail, they can grow a new tail. While they do regenerate that tail, if it's not truly regeneration in an asexual context, because if you only had the tail left from the reptile, it would not be able to regenerate the rest of the reptile, its head and its legs. So kind of keep in mind in an asexual context, the whole organism, regardless of, one piece or half a piece, is able to recreate itself. In plants, you can have vegetative budding or propagation. And in this instance, what you see, the geranium stem is cut off, and then the cutting is placed in soil, and it'll grow new roots in a new plant. So that's an example of vegetative budding. In propagation, what you see 2 organisms at the top, and both of them have a desirable trait and an undesirable trait. The one on the left has those nice big flowers, but a super small root system. The one on the right has small flowers and a large root system. So what can happen is that you can cut the top part of the desirable large flower plant off, and you can graft it into the stem of the large root. And then those two will grow together, and you'll end up with one organism that has nice big flowers and a large root system. So that's another type of asexual reproduction. And then all of the offspring of this plant will have both of those traits as a result. All right, a little bit of general information. Humans have 46 chromosomes, total 23 pairs. And in eukaryotic organisms only, these organisms have small proteins that assist in folding and packaging DNA molecules. DNA is a really big, long molecule. And if we tried to wind that up and fit it into the nucleus, keep in mind we've looked at some of these cells under the microscope and we know they're really small. So the nucleus has to be even smaller. And if we're trying to take something that's as long as your pencil and put it into that nucleus, the tendency of it is it's going to get twisted up and get knotted. So the histones, that's as long into the nucleus, keep packaging DNA. Eukaryotic. A little bit of general information. Humans have 46 chromosomes, total 23 pairs. And in eukaryotic organisms only, these organisms have small proteins that assist in folding and packaging DNA molecules. DNA is a really big, long molecule. And if we tried to wind that up and fit it into the nucleus, keep in mind we've looked at some of these cells under the microscope and we know they're really small. So the nucleus has to be even smaller. And if we're trying to take something that's as long as your pencil and put it into that nucleus, the tendency of it is it's going to get twisted up and get knotted. So the histones assist in helping that DNA from getting tangled up, and it kind of winds around those histone proteins. We will talk more about those, but just kind of a heads up that to help keep DNA organized, eukaryotic organisms have a protein that assists that. And you also know that DNA is copied through the replication process, which happens in the S phase of interphase. Little bit of a difference between plant cells and animal cells. Animal cells will divide by a cell furrow. You've heard me refer to that a couple of times, or a cleavage furrow. And that's kind of happening right here. And you can see it right here as well, where I just highlighted it. And all that happens is the cell membrane just kind of pinches inward until the cytoplasm and the cell are divided into two parts, with a couple that assists that. And you also know that DNA is copied through the replication process, which happens in the S phase of interphase. Little bit of a difference between plant cells and animal cells. Animal cells will divide by a cell furrow. You've heard me refer to that a couple of times, or a cleavage furrow. And that's kind of happening right here. And you can see it right here as well, where I just highlighted it. And all that happens is the cell membrane just kind of pinches inward until the cytoplasm and the cell are divided into two parts. With a plant cell, though, those use a cell plate. And you can just ever so slightly see the cell plate starting to form right here where I'm kind of highlighting. And the cell plate is made using the Golgi apparatus in a plant cell. And it's, again, starting to form right here. It forms from the inside out. So you can see that the cell plate's formed here and it's working its way out. And eventually it divides the cell in half and you end up with two new cells. So again, just to keep in mind, animal cells divide with a cleavage furrow, plant cells divide with a cell plate. All right, some super confusing terms. And we will continue to look at these terms. However, kind of start thinking about how you can visualize these and keep these straight in your mind now. So the first word is chromatin. And the first diagram on the left shows chromatin. All chromatin is a single strand of uncondensed DNA. So and DNA is made up of histones And DNA and those histones, remember, are proteins that the DNA wraps around. The chromatin is uncoiled so that the DNA can be replicated during the S phase of interphase. The only real-time chromatin is going to be present is going to be in interphase, and generally right before and right after DNA replicates. We want that DNA to be open so that it can unzip and be copied. All right, so chromatin is kind of only going to be talked about when we're looking at the S phase of interphase. Right? Now, that chromatin is going to be replicated. And once it's replicated, you are going to end up with this middle picture, and you're going to have two sister chromatids. So this was the original chromatin. This is the copied part of it. And now it's joined together by a centromere. That little dot in the middle is kind of like a piece of Velcro that holds those identical copies of the chromosomes together. Those are going to condense down. All right. And then we're going to call them chromosomes again. So you can see right here, okay, you end up with a sister chromatid and a sister chromatid. The DNA has condensed. That's why it looks all squiggly here. So this is telling you that this has already condensed. Then the reason it does that is because we've got a lot of DNA that we have to get in the nucleus. So each half of this is still considered a sister chromatid as long as they're attached to each other. All right? And together, we call them a chromosome. All right, so another quick look you can take a look at, and there are several pictures that I've included. This is a chromosome. This half is a chromatid. This half is a chromatid. And they're chromatids, excuse me, because again, all of that DNA is coiled and condensed into that chromosome. They're held together by a centromere, right? And once they separate, they'll go back to being chromosomes again. All right. They then can be replicated. And when they replicate, they have to unwind. And when they're unwound, all right, when they're uncondensed, they're called chromatin. So chromatin is uncondensed. Chromatids are condensed and they're with their sisters. All right, so one more view. I've tried to include as many pictures as I can of this so that it kind of helps it make it a little bit easier for you. Here you have a chromosome, and we're using the word chromosome because it's condensed. If it were not condensed, if it were stretched out, it were uncondensed, we would call it chromatin. We have a chromatid here and in its sister, and we still call it one chromosome, but the replicated parts are called chromatids. Right? So lots of words, lots of vocab that goes with cellular reproduction. Unfortunately, a lot of them start with C. And we're going to continue to add words to this. So really think about how you can start getting this vocab as something that you will be able to better understand and visualize moving forward.